All right, so this is actually a little bit different than what that was about. That was about multi-layer brain networks. This is going to be kind of a different world. But I just want to thank all of the organizers for inviting me. I am really interested in individual differences, so I've been so happy to hear all of the work that people have been putting into looking at individual differences in some of these, we'll say, lower scale brain networks. Okay, so as we know, there's all kinds of neuroscience data, right? For me, the micro scale is really going down to neurons, and I know many of you work way below that level here. But there's always kind of this trade-off, at least perhaps until recently, between the spatial resolution and the temporal resolution in all of our neuroimaging data. And so I do a lot of work looking at brain networks, trying to figure out how structure influences function. And I actually work across all of these scales, except today what I'm going to talk about in looking at individual differences is going to be at this macro scale level, where we're looking at MRI data. Okay, and we're going to use, we're specifically here going to be interested in structural brain networks, okay, from DMRI data, and trying to understand how individual differences in these structural networks relate to behavior. I actually have not thought about doing this at the level of the Connectome based on neurons, simply because I didn't know until this week that we had that kind of data. So it's really exciting to see all these new data sets, and maybe at the discussion we can spend some time talking about if any of this is remotely useful to all of you who have this new Connectome data at that level. But for what I've been doing, we've really been limited to looking at these larger scale networks just because that's where we have the full Connectome, or at least it's where I have had access to the full Connectome. So to give you a little idea, at first I'll introduce this idea of personalized brain network models and what they are. And then I'll show you some kind of like proof of principle work that we did using these models to predict task performance in human data. And then I'll close by showing you some methods that we used to try to use these types of models to quantify the types of variability that we see in structural brain networks. All right, so first let's talk about what these things are. So when we're going to build these brain networks, there's a couple of different kinds of networks we can build, right? There's the structural networks, and again, for everything I'm talking about, we're going to be at this larger macro scale level. So we're going to be working with DMRI data, where these structural networks are going to be the density of white matter tracks between brain regions. Okay, so we have our tractography, we have our parcellation of the brain, we turn that into a network, and we get these weighted structural networks, all right? We're also interested in functional networks, though, because from these same types of networks, we can have the nodes being the same brain regions, we can record activity from them, okay, and we can build functional connectivity matrices. For everything I'm going to talk about, we're just going to be using correlations, okay, nothing fancy here. All right, the reason that we're interested in this is there's been a lot of work, trying to say, well, what is the relationship between structure and function? If you have some sort of structural connection, is there a functional connection? And I probably don't have to emphasize this to this audience. A lot of people say, oh, yes, if there's a structural connection, we expect a functional connection, because some of this original work that came out where people saw a correlation between structural connections and functional connections. If you look at this data, you can see that cloud of data points there. Yes, there is a line there, but I think we can all agree that the data doesn't really sit well on that line. Okay, so although there is a statistical correlation, it is by no means exact, and you shouldn't expect a functional connection where you see structure, okay, even in resting state data or anything like that. And I'll comment on this a little bit later. People sometimes try to use these interchangeably, and I want to make a big push that this model will kind of show you why you shouldn't do that. Okay. For anyone who's interested, there's also a really nice review article on this that goes a little more in depth into the relationship between structure and function and shows exactly why it's not always a true relationship, and we have to be a little bit more careful when we try to think about going between these two types of networks. Okay, so what we're going to do to construct these networks is we're going to just build a virtual brain. Okay, we're going to do computational experiments here. So we're going to start with the anatomical network structure, these structural networks that we get from the DMRI data. Okay, so these are going to be our tractography networks. And then instead of actually recording functional data from these brains, we're going to use a mathematical model of dynamics. Okay, so we're going to have some sort of data-driven connectome with the nodes being brain regions. And then we're going to couple mathematical oscillators based on that weighted connectivity. Okay, and so that then we have simulated brain dynamics now, and we can calculate a simulated functional connectivity instead of an observed functional connectivity. All right, and what this does is it allows us to create these virtual brains that we can use to do experiments with that maybe we can't do in a real person. Okay, so just to give you an example, maybe some of you have heard of the virtual brain. This is a software platform for doing this, developed by some colleagues in Europe, and you can use this. We tried to use this actually for our experiments. If you want to make small modifications to the equations that you're using, it ends up being a little bit harder, so we kind of went back to writing all of our own code to do this. But for anyone who's interested and wants to do any of the things that this has been written to do, this is a great platform. You can go in, you can put in your structural data, you can choose what kind of equations you want to run the dynamics, and you can do all kinds of experiments with it, okay? And so people have studied things like TDCS, stroke, epilepsy, all kinds of stuff with this platform. So I just want to make sure that if you want to just kind of dabble in this, this is a really great tool to get started. Okay, so like I said, the big thing about these computational brain networks is we have, from the data, okay, our structural brain network, we're going to combine that with our mathematical model, and we're going to get out these simulated dynamics. So I keep just saying this mathematical model, but what do we actually use? You can use lots of things, and the real answer is it's going to depend on the type of question you want to answer, right? Like everything else does with modeling. What we're using here are just Wilson-Cohen oscillators, okay? So this is a coupled excitatory inhibitory population, okay? And they're kind of in some way representing like cortical columns or something like that. We're not really too concerned with the biological representation here, because what you're going to see is that we're not really using this to get at mechanisms so much as to make predictions, okay? So the important thing about this oscillator model that we've chosen, like I said, it's an excitatory population and inhibitory population. And we're going to model the dynamics, the output here, by looking, this is modeling firing rates, okay? So we're plotting the firing rate of the excitatory population as a function of time. And that's our brain signal that we get out, okay? So these are kind of some examples here. And what I want to point out is depending on the parameters that you put into the model, the system is going to be in one of three states, okay? These are all of these other parameters that I don't really want to point out, okay? But these are just taken from the original Wilson-Cohen model, but it puts the system in a state where you can have three steady state solutions. All right, you have either a low fixed point, an oscillatory state, or a higher fixed point, okay? And that depends on the amount of input going in to this model right here, okay? Whoops, there we go. So this right here, this is maybe what you'll see in some sort of typical coupled system, okay? You'll see some things are kind of like oscillating at a high point, some things are just oscillating, and maybe some things regions don't have any activity at all. But what we can do then is we can go through, and from this activity, we can calculate our observed functional connectivity, all right? When I first started working with these models, okay, let me just give you a little bit of history here. What we were doing is we were actually trying to do a study looking at control theory and trying to identify regions of the brain that could move the brain in different ways, okay? So the advantage of these models is we had a nonlinear model. We could go through, we could stimulate every single region. The IRB doesn't like it if you put in something, you know, where you want to stimulate a person's brain 500 times. It's not considered okay. But we could do that in this model, okay? And so what we had is we had a bunch of brain. So actually, let's see. I think I've actually said most of this already, okay? But that's what we were trying to do, okay? I want to emphasize again, this model has three states, okay? This is for a single oscillator not coupled to anything else, okay? So if it doesn't have as much current coming into it, it's at this low state. If it has some moderate amount of current coming into it, and so we have both like the coupled thing coming from other brain regions, and we have another parameter where we can just flat out input current. So that's our stimulation in this model. Or it can go into this high fixed point, okay? So when I was doing this experiment where I wanted to look at all these people and go through and stimulate their different brain regions, I had data, as it turns out, from 8 people, okay? And I had structural connectomes taken from 3 time points. So we had this nice triplicate data set. This was a while ago. This was one of the first data sets where we actually had structural networks from the same people multiple times. So I put everyone into this model, right? And I go to run it, and I get back my data, and it makes no sense. Like 2 people, yeah.
Sorry, put everyone into this model means that you estimate the parameter from...
So sorry, let me describe better. So in this data set, the only thing that changes in this model, I should have explained this better. You take the structural connectivity, you put it into the model. We actually do not do any parameter fitting of the mathematical model equations. We just leave them all at the same place.
How do you know it's a good set of parameters?
So again, we chose the parameters purely based on the fact that they are from the original Wilson-Cohen paper, and they give you these 3 dynamical states. Okay, you'll see why that's important in a second. But they're not fitting to data. So that's one thing I want to make clear here is the output of this model literally looks something like this. It does not resemble brain activity. Okay, we have oscillators. For these models, what's useful is when you start looking at the functional connectivity, you're correlating, and the relationships between how they oscillate do resemble what we see from actual brain waves. So the actual output here, though, it does not model brain activity. Okay, so we have this triplicate data set. Go to put it in, and the data is just all over the place. It makes no sense. You simulate the same brain region in each people, you look at these functional networks, and it's a disaster. You can't figure out what's going on. Well, it turns out the reason for this is, if you look at this equation here, there's this parameter here. This is a global coupling parameter that just amplifies and controls the amount of input coming in through these connectivity matrices, essentially, right? Just so that it's in the range of the other parameters. So I had tested this on one person, put them in the state so that it was right before their brain would jump into this oscillatory regime, and assumed that that could be done for all of the people. Assuming that these connectomes were very similar. And people who'd used these models before actually tended to not do this for each person, but do it on an average basis. You would just average over the connectomes, and you would put in the average brain and then do these simulations. But we decided, we have these separate ones. We have triplicate scans. Let's do this for each person. So what it turns out is that for each person, if you want them to be in that level right before they jump into that oscillatory state and go up into the limit cycle, you have to choose a different value of this global coupling parameter, okay? And so this is an example here looking at this. Again, we had 8 subjects, 3 scans per subject. So this is the point, basically, where they transition from this inactive to this active state. And if you look at this across 8 people, there's variability within scans within a person, okay? But there's a lot more variability even between people, okay? So this is looking at the interclass correlation right here, a measure of reproducibility. So within subjects, it's very reproducible. Between subjects, it's not, all right? And then this was just another way of trying to look at that, looking at the variance, okay? Yeah.
I'm still a little confused about what you're doing here.
Okay.
So you have all of your individual regions, and each one is tuned to sit just below.
Not each one, sorry. It's a function of all of them. So this is your connectivity matrix.
So how do you
here.
tune the individual regions?
So we don't.
But you have the P parameter.
So right now, we're just setting that to 0.
So you're.
So we're just trying. Yeah. So I'm sorry. So our goal for doing this, I'm sorry, because I'm not really talking about this experiment. I kind of jumped over a bunch of stuff. The goal for doing this was to stimulate brain regions. So we wanted to have them right before they're active, basically, apply a stimulation to them and have them become active. That was and so that was how we ended up normalizing across people, was saying, let's put everyone in that state right before they're active. So that's the only thing that's different between people, is going to be this C5 parameter. Okay, and that's why this is the only thing that's varying. All of the other parameters.
C5 and see where the transition is.
Exactly. And so that way we could kind of normalize across people, because if we didn't, some people when you stimulated, nothing would happen. Some people when you stimulated, nothing would happen, but for a different reason, because they were already at that excited point. And so we really had to normalize across people to be able to do any of this, okay? That was entirely unexpected, all right? This was not something that we thought would happen, because if you looked at these are examples of 4 of those subjects and their structural connectivity matrices. If you look at them, you can see small differences by eye, but if you plot their degree distributions, they're largely the same. The statistical properties of these networks look very similar. It's very hard to take these networks and measure differences between them, okay? I don't remember which ones these are, but these are examples of two different brains over here, okay, shown in a different sense. And if we run them through this model, okay, and we place them like just above the point now where they become active, these are the two patterns of functional connectivity that we get out from two different people. So you start with something that looks very similar, you run it through the model, and what you end up with are these two incredibly different functional networks. Yeah.
And in the model, only that global coupling parameter is different.
Only the global coupling parameter is different. We did not try to fit any of the other parameters. There are lots of people who work with these who do fit the other parameters and try to actually match brain signals. Again, we're not doing that, which is actually a strength of this. The only difference between these two outcomes is the structural connectivity that goes in. Okay, you can imagine a world where you also tried to fit functional parameters, but right now the only difference between these is the input is two slightly different structural connectomes. Okay, so if you look at this, you can actually show that what the model does is it accentuates these differences. So if you look at the variability in these structural networks versus the variability in these functional networks, and this is just the color here is showing the level of variability for each node, you can see that we start with something that's all just kind of like the same, all right, and we end up with something that augments it. We see some that have very low variability and some regions that have high variability. So what we realize is these models are actually really great for studying individual differences. Okay? The harder thing to do with them is normalize across the population. The more fun thing to do is say, okay, let's use these so that we can measure differences that are very hard to measure in our structural networks because the dynamics are going to augment them. Yeah?
So this coupling parameter, to what extent do you think is that a physiological difference, or to what extent could it be a measurement difference?
So, I mean, it's going to be something about the connectome, right? If we go back to this picture here, so this is triplicate data. So for each one of these subjects, we have 3 data points. So there is obviously some measurement error in this, right? You have the same person, you've scanned their brain 3 times, and you do the tractography on it, you get a slightly different network out. And so there is some level of variability there that's just kind of a measurement error.
I was thinking more about something like if you have the white matter and that, I mean, the connectivity strength in the real brain could be different from what is actually measured, right? So that some people have, I don't know, higher diameter white tracts, although their actual physiological connectivity is the same. So more in that direction instead of the measurement.
That is the weights, not so much the connections. Yeah, I haven't really thought about that before. I kind of tend to assume that for things like that, the variability is kind of, for something like that, randomly distributed across the population. And so you would see the same kinds of levels there across a population, and that it would kind of cancel out. But I haven't thought about anything like that. Yeah, that's a good point.
When you say variability, exactly what do you mean?
I knew someone was going to ask me that, and I forgot to look it up. So let's see. So I think this was just based on the degree, okay? And this is the variance across people. And this would then be the variance of the same thing from the functional networks. So I'm pretty sure it was just the degree.
I suppose my question is, like, are you comparing like with like?
Yeah. So this is. I'm pretty sure it's the degree of each region from the structural matrix and from the functional matrix. Okay, so you're summing up the connections of each brain region. Yeah, so in this case, we're looking in this case, I think we had one too with the actual averages, but what we're really asking, right, is about the difference across the population. So we're interested in the variance of that distribution.
Yeah, but I suppose my question is more like, you're looking at variances of different things and kind of saying, there's more variability, say, in the functional than in the structural. Mm-hmm. And there's another measure that's like the coefficient of variation.
In that. So I think these are normalized so that they're in the same range, so that's not
it's probably
yeah. We tend to normalize these networks to be between 0 and 1, so that we're comparing apples
Yeah,
with apples.
that's what that is.
But I'm curious if, instead of this like Wilson-Cowan thing, you would just like a noise-driven linear system that you put one of these connectivity matrices into, would you see similar kind of divergences between the structural and the covariance matrix? Or I'm
So
curious.
that is a good question, because I didn't try that, because originally when we were doing this, we were trying to add nonlinearity. They had done all of it analytically with linear models. And so we didn't even think about that from a terms of individual differences, because we just got excited here and never looked back. But that's a good point. You might be able to see that, but I don't know. Yeah. Okay. All right, so you can use these types of models now to look at a whole lot of different things, all right? So if you have these personalized brain network models, now you're going to have a scan for each individual person, and that's very important that you have data for each individual, and you're going to simulate their brain activity. So one of the things I'm going to talk about today is this idea of stimulation, where you can go in and you can stimulate different regions in the brain, and you can look at the output in this model and ask how that differs again in people. I just want to point out another thing that this has been used for a lot is in the world of epilepsy. All right, so in epilepsy, they do these surgical resections where they actually take out portions of the brain, and it's based on a clinician's identification of where the seizure onset zone is. All right, and it turns out clinicians are about 50-50 at doing a good job on this, and so in about 50% of the cases, the resection works and the patient is seizure-free, and in 50%, it's not. And so this is one of the ways that they've been looking to try to figure out if you can use models like this to predict more correctly where you can do surgical resections. And I believe this has actually gotten to the stage of clinical trials now. Lots of people have different models of doing that one, but I'm pretty sure that this one is actually being used in clinical trials now to do these surgical resections. So there's all kinds of different uses that you can have for these types of models. All right, but what I'm going to talk to you today is a study where we were looking at how stimulating regions across people might be related to task performance. Okay, so, all right, so the data set that we had in this case was we had 10 subjects, and they each performed 3 cognitive tasks, okay? We had the DMRI data for these, so we had their structural network. We did not have any fMRI data while they were doing these tasks, okay? We only had the outcome of the tasks. And this is one of the reasons that when we first did this, we didn't even try to fit to actual brain data because we didn't have any, okay? All we had was the outcome of these tasks. And in this thing, my collaborator was actually, so they did three tasks. They did a verb generation task, a sentence completion task, and a number reading task. And then they would go through and they would stimulate the left inferior frontal gyrus because they thought that this region was associated with the tasks, and then they would have them do the tasks again. Okay? Today I'm not really going to talk about what happens after this because the answer is I don't have a good answer for you. So we're just going to look at this, looking at their structural connectivity, can we predict how they perform on these tasks? Okay? All right, so everything I'm going to talk to from here on out, by the way, basically is done by my former postdoc, Kanika Bansal. She's great. So she came in and she started looking at this data set. And here you see we're looking at the response time. Okay, so low response time means they did well on these tasks. All right, and so we have verb generation, sentence completion, and number reading. And then these different colors here are just doing before and after stim. Okay, the point to get here, though, is that when they did this experiment, there was tons of variability in how people performed, and there was tons of variability in how stimulation affected their performance. So this had just been a pilot study that they were doing. That's why we only have 10 subjects. But they were so confused by this response because it made no sense. They thought that at least in people, stimulating would do the same thing. So we said, okay, well, let's try to see what we can do to study this. Okay, let's see if we can find anything reasonable using these models. So now this is looking across these people and their structural connectivity matrices, and you can see that there are certain connections here that are not very, very variable across people, and there are certain ones that are. Okay, so that's another thing that we noticed in this, is that across people, some of these are very consistent, and some of them are very variable. And we think that that's actually important. All right, so again, just to reiterate, this has these 3 states. What we're going to do is just like before, now we're actually going to go and we're going to kind of normalize again. We're going to take everyone, we're going to set them at that level of coupling that's just before they jump into this oscillatory state because we're interested in what happens now when we go in and we, in our simulations, stimulate the LIFG. Yeah.
In that matrix you showed in the previous slide, how is that sorted? Like, what's that line in the middle mean?
Okay, so this is brain region by brain region. And the way these atlases work, just like the way they're labeled, is the regions are naturally done hemisphere by hemisphere. Okay, so the reason that you see this block-like structure is that you have more connectivity within each brain hemisphere. This is a log scale, by the way, so that you can actually see things. And so then you'll have, actually this one is not a log scale, I'm sorry. When I showed you the structural connectivity ones, that was a log scale. This is just the variability though, so this is looking across these. So this is, but the reason that it ends up having this is because it's the two hemispheres, so they have a very similar structure between right and left, okay?
And the reason it's a matrix is because you're looking at connections between regions.
Right, exactly, because this is a connectome at the end of the day, right? So we're looking at the connections between regions. So this is the variability in that connection strength. Okay. All right, so we're going to normalize this. Well, actually, the first thing we're going to do is we're not going to worry about that. We're going to ask, what is the variability in that transition parameter across our people? Okay? So this is free of any stimulations. We just wanted to say, okay, well, we know that there's variability in these people. And we like to think of this global coupling parameter as some sort of like overall brain excitability, right? Because if you stimulate someone, it's basically telling you how far are they away, how much input does it take to bring them up to that point where they jump into the limit cycle, okay? So people that are very close, it doesn't take much, right? You can have a low global coupling parameter and they're easily going to jump into that, all right? People that you have to really up that parameter, that means you have to put much more input into those people for them to jump in there. So they would kind of have a low natural excitability, okay? So if we looked across our 10 subjects, we actually saw a fair amount of variability in what that parameter was, okay? And so now this is, if we place each of these people right above that transition point, so they're in that limit cycle, this is the model output dynamic. So now we have brain region going down here, and we've just taken a window of time and we've averaged the excitatory activity. Yeah.
I'm a little confused what you mean when you say they're in that limit cycle. I thought it was sort of node by node or region
So
by
it
region.
is, but what happens, so this picture that I'm showing you, this is what happens. Again, I have skipped over all kinds of important things. So I assumed that this would be some sort of slow transition, right? You'd have some nodes that would jump up, and it is not. It's a very abrupt transition. You basically go from nobody being in the limit cycle to everybody being in the limit cycle. It grows a little bit over time, but it is a very sharp transition. So you really can look at it and easily see when it happens because it's a very sharp transition. So that's kind of, it's not going to be every node, but like just even by eye, you can identify where it happens because it'll be like, 50% of them suddenly are in the limit cycle. Okay, so if we put them, I guess this is an example of that too. So you can see that if they were not in this, it would just be a dark blue bar. Every brain region would be at low activity. Okay, but this is showing the average excitatory activity for each of the brain regions. And you can see that for each of these people, when we put them at that right above that point, we see very different patterns of activity across their brain. Okay? Again, this is a very small number of people, right? So this is kind of a proof of principle thing. We've been trying to work on a larger data set to understand more about this. So we asked, well, do we see some sort of correlation with that transition value, so kind of like what we're thinking of as their brain excitability, with how they performed on these cognitive tasks? All right, and the answer was, if we tried to correlate them, we did, but only for the sentence completion task. Okay, we see a significant correlation between the level of that coupling parameter where that transition takes place and how well they performed on the sentence completion task. Okay? So the next thing we was like, okay, well, we see something there. What are the other types of things we can use this model to measure? Well, they were interested in stimulating this LIFG. So what if we go in and we stimulate in these people the LIFG and just look at how the synchronization spreads? Okay, so we used just kind of the dumbest measure you can think of to measure the spread of synchronization, and we averaged over the entire matrix, right? So in this case, we're looking at functional connectivity with correlation. So high correlation, it just means they're synchronized, okay? Two brain regions are synchronized. So if you have a matrix here that's all yellow, it means the whole brain is synchronized, it's oscillating together, all right? So you can kind of look at this visually as where there is yellow, those brain regions are synchronized. Where there is blue, they're not. And we, again, grossly just averaged over the whole thing, okay? Because we weren't really sure what to affect. But that was our hypothesis, was that if you can spread the synchronization, more things can talk to each other more easily or something that might relate to task performance. So we tried to correlate that, and we saw absolutely nothing, okay? If you think about this, though, when I showed you that picture of variability, if I go back a few slides, we have some connections that don't vary across people. We have some that vary a lot. And I tell people I think this is a lot of like a signal to noise type thing. If you are going to look at the whole brain, you're going to have so much noise in your signal that it's going to wash out any effects that you might see. Because if you look at the whole brain, we have some connections that just aren't varying, you know, and that's, you're not going to get enough signal over that background, essentially. So what we did is we went through and we said, okay, my collaborator who's doing these experiments said, but you know, we actually know that there are certain circuits that are involved in these tasks. And specifically, there's one for number reading and there's one for letter reading. Okay, so there's actually some people who had gone through with like electrodes and stimulated different parts of the brain to figure out which ones were involved in these two different types of tasks. And so we lined up our parcellation of the brain with their data from the electrophysiology to get out for our parcellation of the brain which regions were involved in processing alphabets versus processing numbers. And then we said, well, let's not look at the spread of simulation through the brain. Let's look at how it spreads through these circuits that we know are actually involved in these tasks that they're doing. And importantly, differentially involved, right? We have two things that involve words and we have one thing that involves numbers. So when we did that, and we tried to look at this correlation, we saw an effect if we looked at the number reading task and how synchronization spread through this number reading circuit. Okay? Technically, this one is statistically significant, but there's this outlier that we don't trust. Okay? Again, small numbers are a real problem in this study. But in the number reading, we see that if you look at how synchronization spreads through the number reading task, there's a correlation with how they did on the task. So what's interesting about this is these different tasks have different type of cognitive efforts involved in them, okay? And so for certain tasks, we have local involvement, whereas for others, we have global involvement of the brain, okay? So it turns out that for sentence completion, we know that this involves the recruitment of a lot of cognitive systems, okay? It's a global brain task. We saw a correlation when we looked at the relationship between this global parameter of our model, right? This kind of like global excitability. There's a lot of data out there, though, that suggests that number reading is actually a local task. When we read numbers, we do that locally in our brain, okay? And we saw that if we looked within then that circuit and how synchronization spread, we saw the correlation there. So what we really think is that we're able to accentuate these differences between people in these models. But if you want to use this to try to make predictions about tasks or certain things like that, you really have to have a knowledge of what's going on. You have to know something at the level of, you know, where am I stimulating, what tasks are involved, or what regions are involved in the task. Do I have to incorporate other brain regions, things like that. But if you can do that, these can be really useful models, we think, okay? And this is really the interest here that kind of got all of this started was this idea of using stimulation for personalized medicine. Okay, so if you have someone, my collaborator worked on stroke, for example. So if you have someone who comes in with a stroke and you're trying to identify stimulation sites, you know, you could use these models to understand maybe you have different types of people or fall into different categories, and you could use that to look at the effects of stimulation. Okay. All right. So the important thing here is it's we think that it was very promising, okay? We did not lie when we published this. We said this is a proof of principle study, right? You need bigger numbers. So I now have a student who's working with a larger data set, trying to look across this. We think that we might even see like subpopulations in the data of types of people that kind of produce different patterns of connectivity. So maybe we could take in larger data sets and cluster these people into populations. So you have one population that performs similar, another one that's different to that based on these structural connectomes, and how, again, it's very hard to do this at the level of the structural connectivity, but if we instead cluster on the simulated functional networks, we see this distinction. And so in his data set, they actually have these tasks that they're doing with auditory versus visual cues. So we were really interested at how, the auditory circuit would integrate with the visual circuit. And so far, we actually haven't seen interesting things there. But the cerebellum seems to be doing all kinds of stuff in these models. So that was kind of interesting. And we're still working on that. And hopefully, if you talk to me again, I can give you a more complete story about that. But we do seem to see in these larger samples the same types of effects going on, where we can correlate certain types of patterns that we see with certain behaviors, okay, in ways that make sense, too, when you start thinking about the tasks that the people are doing. Okay, so I have a little bit of time left, so I want to kind of like switch a little and talk about another thing that you can use these personalized brain network models for, and that's how to quantify variability, okay? So when I showed you what we were doing before, I said we took the dumbest measure ever, right, and we averaged across these matrices or these subcircuits, okay? But it's very unsophisticated, right? We can do a lot more in terms of trying to actually quantify what's going on in these brain networks. So again, my same postdoc, Kanika, came from a dynamical systems background. And for those in the audience who do that, I'm sure you've a million times had someone ask you, well, how does the brain relate to chimera states? Chimera states, for those who might not be aware of what I'm talking about, the idea here is you have a system of identical coupled oscillators, and for certain regions of parameters, what you can get is states where a subpopulation of the oscillators will be synchronized while the other ones are unsynchronized. Okay, this is an unexpected behavior. Normally, you would expect global synchronization in these networks. And so she was interested in taking some of the tools from chimera theory and applying it to these brain networks to measure the patterns of synchronization that we see, kind of similar to how you would think about chimera networks. Okay? So the way that she did this is she thought about this in a cognitive context. Okay? So we parcellated the brain into these kind of nine standard cognitive systems. Okay, this is a little debatable depending on who you talk about in cognitive science, but these are the ones that we chose. Okay, so now we have this parcellation of the brain. Okay, so she's at a level, I think there's 234 regions in her parcellation. Okay, and each of those is going to be assigned to one of these 9 cognitive systems. Right, this is kind of spatially how they lay out on the brain. And then what she decided to do was to look at the patterns of brain activity that you get from these and classify them as either being a coherent state, a chimera state, or a metastable state, okay, based on the order parameter that you see between the oscillators. So this is where it gets a little confusing. So we've got multiple regions within a single cognitive system. So in what I am showing you here, if you look at the equations here, we're averaging over all of the regions within a cognitive system. And so that's what you can see here, okay? These are system I and system J. And we're actually looking at kind of the average effect of those pairwise synchronizations, okay, by using this order parameter. For interpreting what I'm showing you, again, if it's all yellow, it's all synchronized. Okay, that's going to be our coherent state. We used a threshold of 0.8 here. So if this average order parameter is above 0.8, everywhere it's going to be a coherent state. In a chimera state, you're going to have some cognitive systems that are synchronized and some that aren't, okay? So this is here where you see like some yellow and some of the others. And then you can also have this metastable state where maybe you transiently have some things that are synchronized, but they go in and out, and largely everything just remains unsynchronized, okay? Yeah.
And this is on the simulated.
This is looking at the simulated network.
In the first,
okay?
you explained
So
the
let me
first part.
explain what she did here. This is the next slide. All right, so she had 30 people in this data set. And so she built 30 of these personalized brain networks. So for each subject, now she can go through and she can stimulate every single region in the brain. Okay, so you have two different parameters that we can look across. The simulated brain region, so how does this pattern differ depending on where you stimulate in the brain? And then how does that differ across people? Okay? So she went through and she performed a zillion of these computational experiments. Okay? And here's what she found. Certain stimulation of certain regions tended to produce different types of dominant activity. Okay, remember each of these cognitive systems has multiple regions involved. And so here's looking spatially at the dominant pattern of activity when you simulate these brain regions across people. And what she saw is that regions that are, so here's the midline of the brain right here. If you look at this distance from the midline and you just kind of very grossly bin it to how close it is to the midline of the brain, those close to the brain tend to produce these coherent states, whereas those in the periphery, out by the cortex, tend to give the metastable states. And you can see then the chimera states are kind of everywhere. Okay, so we thought that was interesting. It really seems for these coherent things, these are really driven by these subcortical regions. Okay, they tend to drive these synchronous states, whereas those in the periphery tend to have these more metastable states. Okay, and you can see that there's also a relation with degree of these regions, which makes sense. The higher the degree, the more likely it is to drive synchrony in the brain. But what's important is all of these systems produced chimera states. Okay, so we went through, and you can see this is looking at what percent of the network produced a coherent state, chimera, or metastable state. So these here, these subcortical regions here, are driving the coherent. In the metastable states, this is driven by the cingulopercular network. Okay, for chimera, it's much more evenly distributed. Okay, all of these regions and all these cognitive networks are capable of putting the brain into one of these chimera states. So now I apologize for the next slide that I'm going to show you, because we tried many different ways to put this into something reasonable to visualize, and this was the best we could do. Okay, what's really interesting about this is not that they all, well, it's both interesting, they all produce chimera states, but I told you a chimera state, all that means is that there are some states, some regions that are synchronized and some that aren't, or in our case, some cognitive systems that are synchronized and some that aren't. But you can imagine that which cognitive systems are synchronized matters in the brain, right? We just don't care. If you're someone who does coupled oscillator theory, you don't really care which oscillator is which. You care that some synchronize and some don't. We very much care in the brain which systems synchronize, right? That's very important for brain function. So what I'm showing you here is that this is looking at each of these 9 cognitive systems. And when you stimulate across all of these people and across all of these regions within that, what are the types of patterns you can get of who is synchronized? Okay, so an orange line here is going to be the coherent state, right? All of them are synchronized. A purely white line is going to be the metastable state. Anything where you have that is a mix of orange and white is a chimera state. Okay, and so then this number on the side here is telling you what percentage of the time that happened across all of these stimulations. Remember, we're stimulating multiple brain regions within a system and multiple people. Okay, so there's a lot of differences that you can see here. So like, if you look at, let's see, so here's the subcortical. 44% of the time you get the coherent state. Okay, then you have this other pattern that shows up 15% of the time, the rest of them show up only a few times. All right, if you look at the frontoparietal here, we have, what is that, 42% of the time you get the metastable state, and then very rarely you get these chimera states, okay? But that's very different in things like the motor and somatosensory system. This is much more evenly distributed across this type of states you can get. And I think a nice one here to look at is also the default mode network, mainly because that one's easy to think about in terms of understanding. I guess the motor sensory is too. But the default mode network is kind of the state that you're at before you move to another state. So you want it to be more evenly distributed. You don't want it to always, if it gets some sort of input, go to the same state. You want it to be a little bit more flexible. And you see that it's more evenly distributed across those types of states. And same with the motor and somatosensory, right? You have different types of motion you're making. This makes sense that you get these different types of patterns. Okay? And then here, this is actually just looking at the synchronization probability. So what is the probability that two of these brain regions would be synchronized in any of these states? So you have some with this high level, these dark regions here, that are way more likely to both be in the synchronous state across all of these. Okay, so there are some brain regions that are moving together more than others. All right? And so what we did is we took this and we said, okay, well, let's try to think about this. We have, in that data I'm showing you, we're looking both across brain regions and across people. So let's try to think about it on these two regimes here. So we can have variability across people and across cognitive systems. Okay, and we took all of these cognitive systems now and we classified them based on their robustness. Okay, so what we saw here was that these systems up here, they're going to be stable, relatively stable across individuals and across whichever region we're stimulating within that cognitive system. Okay, so the attention network, and I don't remember what SC stood for, I have to go back. Subcortical, thank you, I should know that one. Right, so these are very consistent, all right? However, down here you have this outlier here, the auditory system. So it's very stable across people, but depending on which region you stimulate in the auditory system, it's very diverse. So what actually was happening here is that she had like two regions that when you stimulated them gave a coherent state and two others that gave a metastable state. Very different types of behavior depending on the region, and that was fairly consistent across people. I gave this talk recently at a cognitive science seminar to a whole bunch of people that work on the auditory system, and they said, well, isn't that just because you can subdivide the auditory system into two subdivisions? And I said, wow, I didn't know that, but that's I have to now go check and see if that actually relates to the way that ours broke down, but it likely could be that this is really just a very poor representation of what the auditory system is actually doing, and it has these two subnetworks within it. Okay, so I think I'm going to stop there and stay on time. So just to summarize, these models are great at accentuating these structural differences. Okay, we really had a hard time measuring differences between people just looking at these structural networks. But as you can see here, we see all kinds of differences when we look at these functional networks derived from doing simulations with them. And our goal, again, in all of this is that hopefully we can use these types of things to develop more personalized medicine. So when you get someone in, create a virtual brain, do some experiments on them, and use that to kind of say, well, you tend to act this way. You'll respond better to this treatment. You tend to be one of these people that acts this other way, and we can better stratify people through approaches like this. So with that, I just want to say thanks. Many of my students who were involved and some of our collaborators that we took this data from. So. And I'm happy to answer questions.